Okay, second time's the charm, I guess. I just got about halfway through this entire PowerPoint and then my recording software stopped responding and crashed, and it didn't save anything I'd recorded. So that's fun. All right, so let's get started with the brain, and I'm going to try and move through this fairly briskly in the hopes that I'll get to a good stopping point and my work all won't be trashed. So, mostly what this particular PowerPoint presentation is about is just the general regions of the brain and their basic functions, some stuff about cerebrospinal fluid and the meninges. So most of what chapter 14 is, besides the cranial nerves, is brain basics. So you may find yourself having lots of questions about more specific or nuanced information about how the brain works. And unfortunately, many of those questions are just going to be sort of beyond the scope of our class. I'm still happy to answer questions about them because it's fun. I like those questions. But, you know, this is a 200 level anatomy class, so we can't really be doing a deep neuroscience dive, unfortunately, as much as I would find that super fun. So let's start with what features and structures and divisions are present in an adult brain, and then we will dial back the clock and go look at what all of this stuff in this picture used to look like, because it didn't always look like this. So we've got, for starters, our cerebrum, which is what most people sort of think of when they think about brain. They're thinking of the pink, wet, wrinkly thing, which is actually just the cerebrum, not even the entirety of the brain. Um, So the cerebrum contains your special sensory processing areas, so visual, auditory, uh, hearing, balance, taste, smell, even your somatic senses. And it also contains your frontal lobe and your limbic system, which is where emotions, memory, higher order cognition, conscious thought processes, and higher order thinking occur, among other things. At the center of the brain, um, enclosed by each cerebral hemisphere, is the diencephalon, which includes the thalamus and the hypothalamus. And that's it for diencephalon. So the thalamus is going to function as a relay center. Basically, it takes incoming afferent sensory information and makes note of where it came from and sends that information to the appropriate location on the cerebral cortex. So we don't get things like pain sensations arriving in the olfactory cortex or visual sensations arriving on the somatosensory cortex, those kinds of things. So we'll investigate more of that thalamic feature and function when we get to the pathways chapter. The hypothalamus is a powerhouse, even though it's pretty small, and among controlling the majority of the endocrine system, it also has centers that regulate basal emotions. So I'm going to add that in. Basal meaning sort of not complex, basic, uh, hardwired, adaptive sort of emotions, things like aggression and anger, rage, and also things like appetitive sexual behavior, libido, etc., as well as some other very basic behaviors and urges and drives like thirst, hunger, those sorts of things. So below that, we have the first portion of the brainstem, which is the midbrain or mesencephalon. And so on the front, we've got the cerebral peduncles, which connect the brainstem to the diencephalon and the cerebrum itself. So what you're seeing here is the cut edge of the peduncle. And then on the back, we have the superior and inferior colliculi. So the rear portions are largely responsible for essentially patterned and automatic motor responses to auditory and visual stimuli. So hearing a noise behind you and jumping and kind of orienting towards it, those sorts of things. And also because there's elements of something called the reticular formation inside the mesencephalon, there is some element of being it being involved in maintenance of consciousness. So the state of you being and staying awake. Under that, we have the pons, which has cerebellar peduncles, so it can take sensory information coming in and relay it both to the cerebellum and upstairs to the thalamus. And it also has a variety of nuclei, so populations of neurons in neural pools and circuits that coordinate both somatic and visceral motor sensors. So an example of one of the somatic motor centers is one that acts on the diaphragm to regulate breathing. And among visceral motor sensors, there's a place called the pontine micturition center, which means it's the nucleus in the pons that coordinates the urination reflex. Inferiorly, we have the medulla oblongata, which is a very fun word to say. This contains a variety of autonomic nuclei as well, which regulate your heart rate, your respiratory rate and depth, coordinates actions of your digestive system, and it also is an important location for descending motor information. So there's these little things called pyramids on the front of it, and that's where descending motor commands cross over to the other side in a step that we call decussation. And finally, we've got the cerebellum. So your cerebellum is basically responsible for checking in with motor patterns as they happen and making sure they're going smoothly. So if you mean to reach for a glass, are you doing so with enough softness to not knock the glass off the countertop, for example? Is your motion smooth? Is it not jerky and dysregulated? So it adjusts output of somatic motor stuff to smooth out what you're doing, and it does a billion other things besides. So we'll talk a little bit about those things as well. Okay, so now we've dialed back the clock and we are looking at what the brain used to look like in each and every one of us. So note that the primary brain vesicles are present at only 3 weeks of development in total. However, I do want to stress at this point, the brain is literally just a hollow tube. It has none of the functions that it will eventually develop. So just because the structure is present embryologically does not mean it works. It absolutely doesn't. So this misconception that many people have is that during embryonic development, the presence of an organ or a structure implies that it is fully functional and working, and that could not be further from the truth. So if you're curious about that, ask me about it. But assertions that are made by certain groups of people about the presence of organs in a developing embryo and what that means about the embryo and its ability to survive or be conscious, those claims are almost never based in reality because they're not coming from developmental biologists. So, do you have a brain that will eventually develop? Yeah, but right now, at 3 weeks, it's just a hollow tube called, well, it used to be the neural tube. Now it's called the neurocole, which is the hollow inside of this. So the neural tube forms after the stage at which we were all a sandwich. So after we were a blastocyst, which is a ball of cells, we kind of flattened out and developed the 3 primary cell layers. So your endoderm, your ectoderm, and your mesoderm. And then once that sandwich was complete, it developed a streak with a groove down the middle of it, and that groove eventually folds up into a tube, becoming the neural tube. So the prosencephalon is eventually going to become the telencephalon, which is the cerebrum once it's all finished with developing. And so as you can see, neither here nor here is it at all wrinkly, like the cerebrum you see at birth. So one of the things that this tube does in order to develop is fold in on itself many, many, many times over as it grows and develops. And that is how we get the wrinkly cerebrum we see today. The mesencephalon of the primary brain vesicles becomes the diencephalon and the midbrain. And the rhombencephalon becomes the metencephalon, which gives rise to the cerebellum and the pons, and the myelencephalon, which gives rise to the medulla oblongata. So again, even at this stage, nothing looks like what it will eventually look like. We can see the structure that will eventually become the cerebellum kind of poking off to the back, and this anterior bulge will become the pons. But really, the majority of embryological brain development is all about folding up. if you fold a neural tissue, you can create room for and the possibility of many more synapses crammed into a small space, which is the space inside the cranium. So, quick note, just because people ask this sometimes and it's interesting to consider, and this is that the correlation between brain size and intelligence isn't very good. There's quite a few creatures on the planet that have larger, heavier brains than we do, and they are not smarter at all. What is more strongly correlated with intelligence and things like decision making, problem solving, tool use, verbal or vocal communication that mean things, etc., is brain wrinkliness. So the wrinklier your brain as an organism, the more likely you are to be very, very intelligent on the higher end as far as animals go. There are some exceptions to this, of course. Another strategy for developing a very, very smart brain, if you are not obligated to have a skull, is you can just kind of distribute your brain throughout your entire body. That's what octopi do. They just have bits of their brain all over the place, and they are very smart to the extent that they can do things like solve problems. So, but for us vertebrates, we have to cram all of it into a skull, so we're a little bit limited in what we can do, and so we just make it real wrinkly. Okay, so the neuracle comes, or excuse me, the neuracle becomes the ventricles and the central canal. So remember from lab, the ventricles are spaces inside of the neural tissue that are filled with cerebrospinal fluid and lined with ciliated ependyma, which is the plural form of ependymal cells. And we'll talk more about that in a moment, but the cilia of the ependymal cells functions to wave back and forth and kind of push the fluid around. So what we can see here is that we have within each cerebral hemisphere, left and right, we have lateral ventricles, which look like a big weird horn. And then we have via the interventricular foramen, which connects the lateral to the third, that cerebrospinal fluid flows down into the third ventricle, which is enclosed on either side by the thalamus, which I'm drawing in here. And then in this picture, this little hole is where the massa intermedia or intermediate mass of the thalamus passes through the third ventricle. So it looks like that. Then passing down through the midbrain, we have the cerebral or midbrain aqueduct, and that widens out to become the fourth ventricle, which is a broad area anterior to the cerebellum and posterior to the pons and the medulla oblongata. And then inferiorly, that narrows into the central canal of the spinal cord. So there are also some apertures here, here, and in the back, which allow the cerebrospinal fluid to not only flow down and around inside of the CNS tissue, but also out into the subarachnoid space, because we want it around our nervous tissue as well as inside of it for a variety of reasons that we will discuss. Oops, go back one. So the same developmental steps are happening for these spaces as well as for the tissue itself. Expands, changes form, it folds up, and becomes this complex series of spaces. So, since we're working up to starting to talk about the production and circulation of cerebrospinal fluid, it's very important that we talk about the Dura Mater. So in my class, we've talked about this in lab already, but it bears repeating. And the first thing I want to mention is that mater is not matter. There's not two T's, there's only one. So it's not pronounced matter, it's pronounced mater, and that means mother. So you've got the tough mother, you've got the spider-like mother, and you've got the soft mother. And these are in order from superficial to deep. So the Dura Mater is very, very tough, structurally sound collagenous sheets, which are kind of like Tyvek. So if you know what Tyvek is, it's used to wrap homes that are being built so that the wood doesn't become waterlogged while the home is waiting to finish being constructed. And it is super puncture resistant and really difficult to tear. So if you took it in between your two hands and you tried to tear it like you would a sheet of paper, can't do it. The Dura Mater is the same in nature as that is. The arachnoid mater is just below that, and it is a thinner connective tissue layer which encloses the subarachnoid space where cerebrospinal fluid circulates. And there's little web-like arachnoid trabeculae which connect the arachnoid mater to the pia mater down below. So these trabeculae have a spider web-like appearance, which gives the arachnoid mater its name. And then the pia mater hugs the surface of the cerebrum and the neural tissue there itself. And considering that our cerebral cortex means the outer layer, and the cortex is made of gray matter. This one is 2 T's, and gray matter equals interneurons, cell bodies, and lots of synapses. Our cerebral cortex is the location where most of our neural processing gets done, and that is what allows us to function. So we want to protect that since it's on the outer side of our brain, not the inside. So we have all these layers to protect us. Quick note before we move on. In most areas of the cerebrum, for example, in the falx cerebri, which partially separates them, and also the covering over of the rest of the cerebral hemispheres, the dura mater is one single layer. But in areas where there is a dural sinus, which is a space that's filled with venous blood, which is oxygen poor, CO2 rich, full of wastes, needs to go back down to get recirculated and go through the kidneys. So the dura mater in these places is separated into the endosteal layer. So endo meaning inside and ost meaning bone. So this is the inside layer that hugs the cranium. And then the meningeal layer, which is opposite the dural sinus space, and it's closer to the arachnoid mater. And so this creates a area that is effectively going to contain that venous blood, and that is where the cerebrospinal fluid eventually drains back into. So, speaking of cerebrospinal fluid, its location is, of course, in the ventricles and the central canal. And also, via those apertures I mentioned, it flows out and surrounds the brain, the cerebellum, and the spinal cord in the subarachnoid space, which I want to note is both present in the cerebrum and in the spinal cord. So, both, not just one. So, there's many functions for the cerebrospinal fluid. We'll talk about them. One of them is to provide an additional layer of physical protection for your cerebrum. So having a skull is great. It's very rigid and hard, and it's difficult to break. But it doesn't stop you from having the possibility of having your head knocked into. And that could create the phenomenon of your cerebrum slamming into the inside of your skull, and you could get a cerebral injury that way. So what we don't want is for that to happen. If the cerebrum and the cerebellum are floating neutrally buoyant in the cerebrospinal fluid, not sitting down on the cranial floor due to gravity or anything like that, then we have this nice liquid cushion around that tissue that protects our own neural tissue from the inside of our skull. So that's the physical aspect. And then the chemical injury is that the cerebrospinal fluid is also there because we can't have our blood cells in our blood directly in contact with our neural tissue. We have to separate them using the blood CSF and blood-brain barrier because, nasties can get into your blood with relative ease, unfortunately, and we don't want any of those nasties near our control center. So instead, we create this fluid that is a proxy for blood and carries wastes away, carries oxygen and carbon dioxide opposite each other, so O2 in, CO2 out, and also carries nutrients like glucose, oops, and amino acids and fatty acids. So omega-3s, for example, you might find those in fish oil, and you might be vaguely familiar with the idea that they're good for neural and brain health. They are. So we have to find a way to get all of those nutrients to the neural tissue without having the blood touch it. So the cerebrospinal fluid is good for that as well. All right, so speaking of the cerebrospinal fluid, let's talk about how it's made. So I'll talk about the volume stuff in a moment, but I think the more important part to lead with is the idea of blood filtrates. So I'm actually going to switch over to whiteboard mode for this, but I prefer the black one. There we go. And draw this out. So this particular idea is not specific to cerebrospinal fluid, but it is an important thing to understand if you are going to understand what cerebrospinal fluid is really all about and other blood filtrates. So I'm just going to take a moment to talk about blood filtrates. So a blood filtrate is any body fluid, and I suddenly can't write that comes from blood. So how does this work? Well, we have capillary endothelial cells, which are squamous, and they're going to just, line the walls of the capillary. Like so. And so you can see that there's going to be red blood cells in here. And then there's plasma, which is mostly water. But also contains things like glucose, which I'll just make be a little pink hexagon because it is actually hexagonal. And you've got ions like chloride. We got good old sodium. So we know that the extracellular fluid in the plasma is rich in sodium. It doesn't have very much potassium, but it does have some. And it has a small amount of dissolved oxygen as well, even though most of the oxygen is inside of the red blood cells, but more on that next term. So if I want to create a body fluid that is watery, one thing I can do is have the pressure inside of a capillary bed be increased somehow, typically by changing the diameter. And that's going to cause stuff to leak out. So water is going to leak out. Oops, no, I don't want this to go away. Go away. So water will leak out, and with that water, so will the ions. And so on and so forth. You get the idea. And then glucose can come along for the ride. And some dissolved oxygen. So the stuff that leaks out does not contain any blood cells. It's like plasma, but no cells, or the large plasma proteins like albumins and stuff. And if you put some more cells around this capillary, you can have the fluid be modified as it leaks out, so you can control what ends up in the fluid and what is kept out. So blood filtrates include cerebrospinal fluid, aqueous humor in the front of your eyeball, tears, sweat, urine, all kinds of stuff. So this is a common motif for how we make watery fluids in our body. We just pull them out of our blood. Oh, I forgot one important one. Gastric juice, pancreatic juice, saliva. So as you can see, the list goes on and on. So we're going to touch base with all of these fluids over the course of the 2 terms that we're doing A and P together. So I figured it would be a good idea, and we might as well just talk about blood filtrates right off the bat. So cerebrospinal fluid is one of those. So now, let's talk about that. So filtration happens at a structure called the choroid plexus, which are specialized capillaries covered by ependymal cells. And the reason they're covered by ependymal cells is because we have little tight junctions between the ependymal cells, which prevent things from sneaking between the cells. So as fluid leaks out of this capillary, so the blood's circulating through, passing through, but it's physically separated from the cerebrospinal fluid, useful things can leak out, and they're assisted in doing so by the ependymal cells. But things that we want to have stay in here will stay in due to those tight junctions and some other features of ependymal cells. So that allows us to make cerebrospinal fluid, but make sure that we're not including in there anything that we don't want. And it also helps with removal of waste. So neurons are very metabolically busy cells. They produce a lot of waste products like ammonia, urea, other nitrogenous wastes, fatty acids, metabolites of various processes, including neurotransmitter metabolites, all kinds of stuff that we have to get rid of. We don't want them building up in our synapses and impairing our neural function. So those all get dumped into the capillaries and carried into the blood so that they can get filtered out by the kidneys. And over here we have the astrocyte feet forming the same function around capillaries that are in the neural tissue or on it. And so these are going to do the same thing where the astrocyte feet just very tightly control what can escape the capillary and what has to stay in. And between those two, we have a really effective barrier between our blood and our central nervous system tissue, which is great. So these little choroid plexuses are located on our ventricular walls. So these fluid-filled spaces, each of them have lateral, third, and fourth have their own choroid plexus, and they're all producing cerebrospinal fluid. Now, you produce about 1/2 liter of CSF per day, but that's not how much you have in you at any given time. You only have about 150 milliliters at any one time, and that's because it is filtered out at the same rate, ideally, as it is reabsorbed. Assuming that filtration and reabsorption are matched in rates, you should have consistently this number, but it's not the same 150 milliliters all day. So it's replaced about every 8 hours, and the rate of turnover increases during sleep. So one of the reasons that if you prevent somebody from sleeping, they die is because you don't have that increase in cerebrospinal fluid circulation, and waste builds up between your neurons, and eventually you get synaptic failure because there's just gunk floating around between your neurons. So again, protect your sleep. It is very important that you do so. Okay, so let's talk about reabsorption. So it circulates around, so it's created in the lateral ventricles, then it flows into the third ventricle, From there, it flows down the cerebral aqueduct into the fourth ventricle and out via the posterior and lateral apertures to occupy the subarachnoid space. And part of it is the cilia, and part of it is just pressure differences in the system. So because liquid is incompressible, if I'm making new liquid, that just pushes the old liquid out of the way, and that pushes older CSF further down the line. So in places where there are dural sinuses, we've got this situation where there is a endosteal layer of the dura mater, and then there's a fluid-filled space which is full of venous blood, and then there is the meningeal layer, and poking up through the meningeal layer into the sinus itself is this extension of the arachnoid mater called arachnoid granulations or villi. And this produces a thinner membrane through which the cerebrospinal fluid can enter the venous circulation. All of the dural sinuses, also called dural venous sinuses, converge on each other inferiorly to drain into the jugular vein, and that takes the blood back through your heart and into your circulation, so all of the wastes that it's carrying will end up being filtered out by your kidneys. So your CSF, you end up peeing it out. That's assuming that your circulation and production rate matches reabsorption. There is consequences if it doesn't. And this is an example of one such consequence. So the term hydrocephalus, which is also sometimes called water on the brain. So hydro means water, head. So this picture is sad. Yes, it's also medically interesting. So this is a transilluminated neonatal cranium. So transilluminated is just like candling an egg, you just shine a light through it. So you can see the very expanded lateral ventricles here, along with the cerebral vasculature, because the buildup of fluid pushes on the cranial bones and makes them thin and fragile and expands the cranial size and volume unless it's treated. So this is what happens if reabsorption is blocked, and so formation is outpacing reabsorption. So if the arachnoid granulations just don't form properly for some reason. The modern solution to hydrocephalus is a shunt, which basically it's placed surgically very, very early in the infant's life, so it's scary surgery. But basically it provides an outlet for the cerebrospinal fluid to go somewhere instead of building up, and typically that somewhere is into the abdomen. So it just goes into the abdomen and gets reabsorbed across, the abdominal surfaces. It combines with the peritoneal fluid, and then it ends up filtering out, and everything's fine. So that's, of course, assuming that you have access to developed nation level medical care. Not everyone does. So let's just take a moment to be grateful for the privilege of having access to the possibility of that surgery. Okay, so the blood-brain barrier. As I mentioned previously, unfortunately, it's relatively easy for nasties to get into our blood. So for example, let's say I get a pretty gnarly cut from falling down, or like getting a bad splinter when I'm chopping wood or something like that, and there's bacteria that hitch a ride from my skin into the wound, and they weasel their way into my blood. If there was direct contact between my central nervous system tissue and my blood, it would be no problem at all for those bacteria to go infect my brain and create inflammation and kill me. Because the brain is enclosed in a cranium, even a very small amount of brain inflammation can kill you by creating increases in intracranial pressure, which basically ruin your brain function. So we want to avoid that at all costs. So we have this set of redundant systems that collectively we call the blood-brain barrier, which basically creates a situation where what makes it into your cerebrospinal fluid is very tightly controlled. So tight junctions is one. So Nothing can sneak between cells. So it's when cell A and cell B over here are sort of quilted together. So when you make a quilt, you take two sheets of fabric and put something between them and then sew the fabric so that they're joined across the batting in the middle. Tight junctions are the same. So this means that, let's say there's a little bacterium here. And he's like, yeah, I want to go in there. He tries, boom, denied, can't go through. So anything that might try and sneak between your cells, not possible. Astrocytes also influence capillary permeability. So you can see that their little feet are wrapping around this capillary, and then the feet are joined to each other with tight junctions. And so astrocytes permit things to enter and exit the blood via a process called transcytosis, where if we have, here's an astrocyte foot, here is the cerebrospinal fluid, and here is blood. Actually, let's call this just neural tissue. So this is an astrocyte foot. So transcytosis is where there's endocytosis on one side, and then the vesicle just goes right on across, and there's exocytosis on the other side. So any nutritive molecules or anything that might need to get transported to the neural tissue can go through the astrocyte foot like that, but the astrocyte gets to decide what crosses and what does not. So that's kind of the deal there. So there are a couple areas in which the blood-brain barrier is not complete, meaning it's not completely absent, it's just not fully and robustly in place. So at the capillaries of the choroid plexus itself, we do call those the blood CSF barrier. The tight junctions of the ependymal cells take care of that very, very competently, but it doesn't also include astrocyte feet, so we don't consider it to be complete there. And the other areas are, of course, the areas of the brain that are also endocrine. So endocrine glands perform their function by creating signaling chemicals and dumping those chemicals into the blood. So because the endocrine structures of our brain need access to our blood to do their jobs, there are some weakened areas of the blood-brain barrier in those spots so that they can do things like put melatonin or serotonin into our bloodstream so that those hormones can perform their functions. So even though it's really great to have a blood-brain barrier, it is necessary in some spots to have it be a little bit more relaxed than it would otherwise be. All right, so I think that's long enough for that first part of the video. In the next video, we'll talk about the regions of the brainstem and what each of them do. But for now, I'm going to end this and go get this posted so that you guys have a bunch of new content to look at. And thank you for your attention as always. I will see you in the next video. Bye-bye.